Bismillahirrahmanirrahim. Assalamu alaikum. My name is Mahnoor Tanveer. My student ID is BC. I am from Virtual University of Pakistan. My course code is bt-602, which is Seminar 2. I have been given a task of video presentation on the topic of flow cytometry. My topic of presentation is flow cytometry. The table of contents of flow cytometry are: what is flow cytometry, components in flow cytometry, parameter in analysis such as forward scatter cytometry and side scatter cytometry, data representation, common technologies in flow cytometers, common application in flow cytometry. First of all, I'll discuss flow cytometry. What is flow cytometry? The rapid measurement of particles of fluorescent flowing in a stream is called flow cytometry. Why we use flow cytometry? The purpose of flow cytometry is to count cells. Viable count means how many cells are alive or are not. Cell cycle, which tell us about in which phase cells are lying, mean G1 phase, G2 phase, S phase, etc. Flow cytometry is also used to detect the cell's health, such as apoptosis and cancer research. It also used in any immunological markers such as CD4 and CD8, etc. The range which can measure the flow cytometry is 0.0 micrometer to 100 micrometers. This is a diagram of flow cytometry which describes us how the flow cytometry works. Flow cytometer components. Flow cytometer basically composed of four basic components. Number 1, fluidic system. Number 2, laser. Number 3, optics. And number 4, electronic or external computer system. A strong device can be added to collect cells after they have run past the laser. Here is a diagram of flow cytometry components which give us a brief description. As we can see that in the fluidic system, when the cells passing through the laser light emit, when the cells hit the laser, it can go to the optics and detect by the electronics, and electronics pass through it to the computer device, and we can see the result through the software in the different graphs form. Are you measuring? We are measuring two things. Number 1, fluorescent intensity, and number 2, light scatter intensity. In fluorescent intensity, light emitted by fluorescent dyes 522 nanometer emission means fluorescent intensity. It can measure the small amount of fluorescence emitted by the cells. In light scatter intensity, it consists of two more parts, which is forward scatter intensity and side scatter. In the case of light scatter, the interaction of the light with the cells. Side scatter tells us about the complexity of the cells, whereas forward scatter tells us about the size of the cell. Data display. After the process, we can collect the data in a different form, such as reducing raw data to pictures. We use histogram. In the case of frequency distribution of digitalized intensity values for a single parameter. Dot plots. Used for it, plots of two parameters. Each dot represents one cell. The intensities of two parameters plotted as X and Y distribution. Cytometer technology. Where the flow cytometer technology is used? In the case of conventional flow cytometry, it used to count cells and their fluorescence. Number 2. Cell sorter. In the case of cell sorter, everything similar to conventional flow cytometer, but it used to separate cells on the basis of fluorescent signals. We receive quantified number of cells in tubes, and the cells can be used for downstream application. Number 3. Imaging flow cytometer. For that, it takes images 60x or 40x images of every cells. Everything similar to conventional flow cytometers technologies in this case also. Flow cytometer application. There is a wide range where the flow cytometer are used, such as in clinical application. Flow cytometer used for HIV, CD4 and CD8 ratio for disease progression. In blood related disorders, especially cancers and research applications. Flow cytometer application is also cell health assay. It's used in apoptosis, cell cycle, immunophenotyping, cell signaling pathway, any surface markers, ploidy analysis in plants. Why we use flow cytometer? Because of the three things, which are speed, precision and sensitivity. Speed means it can use for a large amount of the data, means large amount of the cells. We can use a flow cytometer to run a lot of cells. Precision means it resolves small difference among cells, means if a small fluorescence is emitted, it can detect it. Sensitivity, it can detect very weak fluorescence from cells. These are the three advantages of flow cytometer usage. The references where I collect the data for my presentation, which are lebomb.com, Wikipedia.org, PubMed NCBI, and ScienceDirect.com. Thank you. Allah Hafiz.